Test, test. interest for our workshop. So far we have 35 people connected, but the interest was very strong. That's why we had to use YouTube, because we surpassed the limit of 100 participants for Zoom and Google Meets, and thank you very much for that. It's our first time doing YouTube streams, so if something goes wrong, you can just tell us in the live chat.
Yes, let us know if you can hear us well. Hopefully.
Introduce ourselves, maybe?
Yes. So I'm Roman Makarov. I'm a PhD candidate in Biophysical. I study neurons. I create biophysical models, and as part of my PhD, I was developing the DendroTweaks toolbox, which we'll see today.
Okay, my name is Michalis Pagalos, and I recently finished my PhD also in the Polarized lab. During my PhD, I developed Dendrify, which is a tool to develop simplified SNNs that capture dendritic properties. And today we're going to both describe our tools that we kind of jointly developed during our PhDs in the Polarized lab. And here you can see where we can find the publications. Ramon?
Yes, so today our workshop will be organized in two parts. The first part will be about biophysical modeling. We will see how to work with the DendroTweaks toolbox to build single-cell biophysical models with active dendrites.
And as I mentioned, Dendrify is the second part of the workshop, where we're going to see together some code on how to develop simplified dendritic networks in Brian 2.
Yes, so here you can see the program of the workshop. Now we have the introduction. I think it will be shorter than 30 minutes. Then we will start with the first part. It will have an introduction with some slides, and then a hands-on session where we will be working on some tutorials, and later we will have a Q&A session. Yes, and the Q&A session will be in Google Meets. Hopefully you have the link. We will also share it later. And then we'll have a lunch break for 1 hour between the two parts.
Yes, then again we'll talk about the Dendrify, and we'll have a similar structure, an introduction in the beginning, some exercises, and then Dendrify Q&A, and then the closing remarks. So this entire workshop is going to be recorded, right?
Yes.
For the hardcores that they want to join both parts, they can start now. We're going to be very precise in the times, so we're going to respect your time. Otherwise, you can just choose what you prefer. You can just join DendroTweaks or Dendrify later. But if you join live, you have the advantage to use the live chat to ask questions on the go, and the Q&A later. So it's highly recommended for someone that is not familiar with these tools, and they like to be, they like more interactive tutorials. We also like more interactive tutorials. So are we starting?
Yes.
Okay, the floor is yours. I'm going to be a spectator for the next hour, so see ya.
Thanks. Okay, so now we're ready to begin with the first part, where we'll talk about biophysical models and the DendroTweaks toolbox. So first of all, why do we even want to create models? So the general principle that I like to follow is that what I can build, I can understand. The models, usually when we create the models, we want to reproduce some phenomena observed in experiments, and we want to explain the underlying mechanisms of this phenomenon. And the models specifically, we can control all the parameters, all the variables in our models, which is not possible in experiments, and we can perform some manipulations which are not possible in biological tissue. So the brain is a multi-scale system, and we have to keep that in mind even when modeling single neurons, because we have to take into account both the subcellular properties, such as properties of the ion channels, but at the same time, even when we model a single neuron, we have to think about a network in which this neuron is located, because we have to model, for example, the inputs that it receives from other neurons in this network. So, when developing our models, we have to keep in mind this multi-scale organization of the brain. And another thing that we need to keep in mind is that we can create our models at different levels of detail. So here you can see two famous models. The leak integrating fire model, when usually you assume that a neuron is a single compartment, isopotential compartment, and you model its behavior with a simple reset mechanism. So you model the behavior of voltage, of memory voltage. The other model is the Hodgkin-Huxley model, where you take into account also kinetics of ion channels in the dendrite and in the neuron and in the dendrites. So usually these cells are multi-compartmental, and this part of the workshop will be about these more mechanistic models, where we want to explicitly take into account channel kinetics, morphology of neurons. So we go to a higher level of detail. Yes, so as I mentioned, because dendrites have this complex structure, complex morphology, we cannot just ignore them and assume that neurons can be modeled as a single compartment. So we can include this information also in our models. And also it is important to mention that there are multiple levels of detailization, even when you want to take dendrites into account. So you can use something as simple as a so-called ball and stick model. Yes, or we can take the morphology from the.
Sorry to interrupt. The sound is a bit low.
So.
Or put on the headphones so that the microphone is closer to your mouth, maybe.
Okay, sorry. I hope now you can hear me better.
Put this closer.
So the take-home message of this slide is that we can model neurons at different levels of abstraction, at different levels of complexity. We can include as many details as we want. And of course it's a trade-off, because more details give us better accuracy, but at the same time they are more complex to optimize and create. So the state of the art models, if we speak about detailed biophysical models, are the models of cortical columns. For example, this one from the also similar works for the somatosensory cortex and for the motor cortex. So these models, they take the whole complexity of dendritic morphology into account. However, not all dendritic properties are present even in these complex models. So from the experimental work, we know that dendrites are not just passive cables that conduct the synaptic inputs to the soma. There are multiple types of dendritic events, such as calcium spikes, sodium spikes, and NMDA spikes. So these are different types of events similar to somatic action potentials, which are mediated by various ion channels. And they can be either local, constrained to specific branches, or they can be global and propagate from one part of the dendritic tree to the other. So from the dendrites to the soma, or from the soma to the dendrites. And this could result in nonlinear integration of synaptic inputs. So the idea here is that because of these properties of dendrites, because of the presence of various ion channels in the dendrites, the synaptic potentials do not just sum linearly, but they can either result in sublinear summation or sublinear summation. And finally, these properties can change the way the neuron responds to a given input partner. So for example, if we place the same synaptic inputs with the same activation times, same kinetic properties, but we distribute them differently. So in one case, we just put them everywhere in the dendritic tree, and in the other case, we place them in specific branches. As you can see here, the response will be completely different. So in one case, we have a very little number of spikes. In the other case, the cell is bursting with multiple spikes. So that means that dendrites are important for integration of synaptic inputs that come from other neurons in the network. And we have to take them into account. So we have to model them and understand how they work. And specifically for these, I developed this toolbox, which is called DendroTweaks. It's a Python toolbox for developing single cell models with active dendrites. Active dendrites means the dendrites that have ion channels and that can generate this complex activity that we discussed before. So with DendroTweaks, you can do many things. You can work with the morphology of your neuron. You can work with ion channels, with kinetic properties of these channels, and with the distribution of these channels. And you can run simulations inside the toolbox and analyze the activity later. Okay, so there are two ways to use DendroTweaks. So it's a Python package, so you just can install it as any other package using pip install, for example. Or you can use the graphical interface. You can use the graphical interface, which is available at our online platform, DendroTweaks.dendrites.gr. And of course, you can use it locally on your machine if you want. So, speaking about neuronal modeling, if you want to create a model of a single neuron, the three key components that you need is the morphology, the biophysical properties of different ion channels, for example, and you need the stimulation protocol. So usually we either place synaptic inputs to our neuron to activate it, or we inject some current, as people do in experiments. And the structure of a model in DendroTweaks follows this idea of these three components. So usually when you create the model in DendroTweaks, you create a specific folder, and inside you have subfolders for morphology, for biophysical properties, and for the stimulation protocols. And yes, once we have this structure, we can run the model in a simulator. So the default simulator that is used in DendroTweaks is Neuron. There are also some functionalities that support Jaxley, but the general idea of DendroTweaks is to make it simulator agnostic, so you create just a high-level representation of your neuron, and then you run simulations using some external software. So the general idea is to separate the parameters of your model from the code that runs the simulation. And once we create our model in DendroTweaks, we end up with such a representation. So the first component of a model is the morphology. We have neurons have very diverse morphology, as you can see here, in various brain regions and in various species. The morphology is quite different, but even in the cortex, if you look at different layers and different cell types, you can see that morphology is different. So we need to take this into account. Now we have access to detailed images from electron microscopy that shows us the full complexity of dendritic morphology. But usually in our models, we don't take all these details into account, so we don't model, for example, specific dendritic spines. Usually what we do is the following. So we have this very popular format to represent neuronal morphologies, which is called SWC. And as you can see here, we basically have a file with multiple points in 3D space. So we place these points along the dendrite, for example, and each point has its index, its type, for example, whether it is the soma or an axon, a dendrite. Then it has 3 coordinates and the radius associated with this point, as you can see here. And also we know the parent of this point, the index of the point, let's say, to which a given point is connected. So that's how we represent dendritic morphology. And there is a database that contains various dendritic morphologies. You can find them easily following this link. So usually when we talk about the morphology, we also need to take into account the domains, the functional or morphological domains of our cell, and typically we would speak about the soma, the axon, and the dendrites, but the dendrites could be also subdivided into various types. So for example, for a pyramidal cell in the neocortex, we can differentiate the basal dendrites, we can differentiate the apical dendrite, which can be subdivided to the tuft, the trunk, and the oblique dendrites. So the points that we created earlier in our SWC files can be combined to the sections. So the sections are the parts of the morphology between the bifurcation points. So for example, these points form a section, and together the sections can be combined into domains. So a domain in the end is a collection of sections. And then when we want to run the actual simulation, we subdivide sections into segments. So we do it because we cannot run a simulation for a continuous system. We need to discretize our model in space and time, and to discretize it in space, we subdivide it into segments. And then we can think about each segment as an electrical circuit, equivalent electrical circuit, which is also called RC circuit, which has a capacitor and resistors. We'll see this a bit later. So now I want to emphasize how we represent morphologies in DendroTweaks. So we have this point tree created from an SWC file. Then, so basically we use graphs to represent different parts of morphology. And we create this point tree graph using the points from the SWC file. Then we combine the points into sections, and we create the graph where now each node of the graph is a section. And then we break down the sections into segments in order to run a simulation. In general, we can think about models as network graphs. So you can notice that it's the same idea applied at different levels. So for the segments, we have the graph that represents segments, and we can think of groups of these segments, and for the sections, the groups will be the domains. If we go to the network level, now each node is a cell, and the groups are populations. So this idea can be applied at different levels. So now let's talk about the biophysical properties. And usually when we talk about biophysical properties, the first thing that comes to our mind is ion channels, because basically the ion channels define all the interesting properties of neuronal membrane, such as the dynamics of action potentials. And as you can see here, these structures are transmembrane proteins, and they have a very complex molecular structure, but in biophysical models, usually we don't take this structure into account explicitly. And what we want really to model is the kinetics of this channel. So basically we want to know how much current flows through a given channel. So yes, there is a great variety of ion channels which can be subdivided, for example, by the type of ions. So there are sodium channels, potassium channels, calcium channels, and then there are different subtypes which have different properties, and we can create a model for each of these subtypes. So for example, here you can see the classification of potassium channels, which is commonly used in biophysical modeling. So usually we need to incorporate in our models these voltage-gated potassium channels, which can have different kinetics. So they could be fast, they could be slow, and also they can have inactivation or not have it. And in the end, usually in many models that you can find online, you will find these 4 types of channels. So yes, it's good to know this classification because it helps you to understand what channels are present in your model. So as I said before, we usually model neuronal membranes as electrical circuits. So the membrane itself in the circuit works as a capacitor. It can collect the charge, can accumulate the charge, and the ion channels, they work as resistors. So resistors, or sometimes they are also called conductances, and some of these conductances can vary with the membrane potential, and some of them remain open all the time. Such as the conductances of the lead channels. Okay, so basically here you can see the equation that dictates the behavior of the membrane potential. So we have our membrane potential here, and basically this equation shows us that the sum of all currents at a given component should be equal to 0, or yes, and now we are interested in this term here, which is the sum of channel currents. So for a given channel, the current is defined as the channel conductance times the driving force. So the driving force is the difference between the membrane potential at a given moment of time, and the equilibrium potential of the ion. Yes, and now we can decompose the conductance from the previous equation into the maximal channel conductance, or sometimes it serves as a proxy to the channel density in a given part of the membrane. And the probability function, which defines the opening probability of this channel. And usually it depends on various factors. The most common one are the voltage of the membrane itself when we talk about voltage gated ion channels. So this this function usually depends on multiple state variables. And these state variables basically is the part of our model which defines the channel kinetics. So how the channel open and close. Okay, I think we will skip this part for now, but what I wanted to show, basically, if we take a look, for example, at the voltage gated sodium channel, here you can see these state variables. So this is a famous equation from the Hodgkin-Huxley model, and basically we can see here how these two state variables interact. So basically now also what we want to know is how each of these variables evolves based on the membrane potential. And we have these two curves, inactivation and activation curve. So the activation curve tells us how many channels are open given that the membrane potential has, for example, the value of minus 50 millivolts. So for this specific channel, this probability to be open is low at minus 50 millivolts. But if we go to 0 millivolts, already you can see that the probability to be open is close to 1. So most of the channels will be open. And for the inactivation curve, it's the opposite. So most of the channels will be inactive at so this curve tells you the fraction of channels that will be inactivated depending on the membrane potential. And also this plot here shows us the time constants of these channels. So basically how fast are these changes, this opening, inactivation, and so on. And to model a channel, basically we need to reproduce these curves. And we can do this using models written in N-model language. So sometimes they are called mod files. And basically here we specify the equations that we have seen so far in the previous slides. And we specify some parameters of the channels, and what we get in the end is the kinetic curves that I showed you on the previous slide, and finally we get the current that enters the cell, for example, through this specific type of channels. Okay, so once we have this kinetic model, we want also to define the distributions of channels, because usually these distributions are not uniform. So for example, you can have a higher density of channels in the distal part of the dendritic tree, or there could be a higher density of channels at the soma. So you have to answer the two questions, basically, where you want to distribute your channels and how you want to do it. So in DendroTweaks, to understand where you want to place your channels, you should specify the groups of segments. So we discussed the segments before. Now we can just select a specific group of this segment tree, and we can say that for these specific segments, which are part of this group, we want to use a distribution function, and we want to take the values of our parameter based on this distribution function. Okay, and here I also want to mention the difference between domains and groups. They will become more evident later when we will go through the tutorial. But the general idea is that the domains are collections of sections. So and segment groups can include multiple domains, for example, or they can align with a given domain, or they can select only specific parts. For example, if we want to have a higher density of ion channels in the distal part of the dendritic tree, we can create a specific group only for that, and we don't have to include the entire dendritic tuft, for example.
We have a question from the chat.
Okay, we'll have like a 5-minute break before we start. Okay, and basically we define the groups, and we assign to each group a distribution function, which could be any type of function we want. So it could be a constant distribution, it could be a linear decrease or increase. So basically, by combining all these types of distributions, we can match the experimental data about the densities of ion channels in our neuron. Okay, and now so we have covered the two components of our model: morphology, biophysical properties. Now we go to the stimuli, because now we want actually to activate our cell and produce some activity. And the natural way to do it is to place some synapses on our cell. And again, real biological synapses are very complex systems with multiple proteins and other molecules that interact. As we did for ion channels, we don't want to take into account all this complexity. Usually, we want again to model only the current that is entering the cell through a given synapse. And the equation is basically identical to the one that we saw before for the ion channels, but in this case we model the conductance of a synapse. And there are multiple ways to do so. So basically what we want to reproduce here is something similar to the postsynaptic potentials. So we want to reproduce this effect of a synapse on the postsynaptic membrane. Usually we model our conductance to follow this stereotypical shape. That is going to produce the postsynaptic potentials in our cell. Okay. Yes, and when we create single cell models, usually we don't model explicitly the network, the surrounding network. We create so-called virtual populations of presynaptic neurons. So we model explicitly only our postsynaptic cell, and for the populations of presynaptic neurons, we just define the synaptic properties, and we define the activation times of these presynaptic cells. Okay, so once we have all these three components together, we can run a simulation, and we can run various validation protocols to make sure that our cell behaves as a real biological cell. So we can, for example, inject a current, as people usually do in experiments using patch clamp. We can inject the current and produce somatic spikes, measure the properties of the spikes and compare them to experimental recordings, and we can use various protocols to validate dendritic activity. So we can validate, for example, voltage attenuation, synaptic integration in dendrites, and so on. So all these validation protocols are readily available in DendroTweaks, so you can use them out of the box basically, and we will see in the hands-on part how we can do this. Okay, so this was the introduction. Now we will move on to the hands-on part, where we will take a look at some tutorials. I suggest now to take a short break, 5 minutes maybe, and then we will be back to take a look at the tutorials. Yes, in the meantime, if you have some questions for this part, you can write them in the chat, or you can keep them for the Q&A session. hear me. So, before we start with the tutorials, I will quickly answer the questions that I see in the chat. So we have one question about why, like what causes the currents in the RC circuit to zero. So basically this is the law that in an electrical circuit, in each node of the electrical circuit, the currents should sum up to zero. It's called the Kirchhoff law. I'll send later probably a link or something in the chat with more details on this. And we can discuss it maybe during the Q&A in more detail. So I also see another question, which book I would recommend to start with this topic. So there is this nice book which is called Principles of Computational Modeling in Neuroscience. I will also share a link later. Okay, there's another question. So if there is some distance between nearby segments. So basically, when we discretize our models, when we create these segments, so our spatial resolution is defined by these segments. So basically all synapses which come to a single segment, they are not differentiated in space. But of course, there is a distance between nearby segments. So if you have two synapses in different segments, there will be a distance between them. Okay, and there is one more question about the distance function for the distributions, I think. These functions, they give us basically the value of a parameter. So for example, we want to set, let's say, conductance of a sodium channel in our model. And we want to say first that for some reason it should increase with the distance from the zone. So we take a linear function which is increasing as a function of the distance basically, and based on the value of this function for the specific segment, we assign the conductance of our sodium channel. So these values do not depend on the properties of the electrical circuit. I'm not sure if this was the question. We can also discuss it in more details during the Q&A section. But yes, the general idea is that these functions, usually they depend only on the distance from the soma, and they give us a specific value that we want to assign to a parameter in our model. Okay, we'll have also more time during the Q&A session, and now I would like to move on, and we'll start with the tutorials. So you can see here our online platform. So one option to use DendroTweaks use it online. If we click this launch button, then we can open the application in our browser and use a graphical interface to work with our models. But what I want to do now is to follow the tutorials that we have in our documentation, so we can see how the toolbox actually works, what kind of code do we need to run to create our model. So you can so first of all, if you want to follow these tutorials, follow along, you can go to our website DendroTweaks.dendrites.gr, and then you can open the documentation page here, and we will start with the quick start tutorial to see the key components of a model. So this tutorial, you can open it in Google Colabs. If you click this button here, open in Colab. Okay, I'll also grab a link just in case. Chat. Okay. So first of all, we need to install the DendroTweaks package. Just with the pip install command, pip install DendroTweaks, and then we also need to import some other libraries such as NumPy, Matplotlib for some calculations and plotting. Here we just check the version, and we just modify the theme for the plots here. Okay, so we will download some examples from the repository. Now you can see here we have a folder with examples, and as I mentioned before, for each model we create a folder, and within this folder we have subfolders for biophysics, which includes the channel models as mod files. So we have a sodium channel, for example. Here we have several potassium channels, and we have several calcium channels for this specific model. Also, we have these JSON files. They define the configuration of our model. So we will learn how to create these files in this tutorial. And in the morphology folder we have SWC files. If we open one of these files, again we see the structure that I showed you earlier, with the types, coordinates, etc. And in the Stimulate folder we have different subfolders again, which define various experimental protocols, let's say. So we have one protocol which will inject depolarizing current at the soma. We have another protocol, let's say, that will place 50 synapses in the dendritic tree. So all these protocols, all these files were created beforehand as an example, but what you really need to start developing your model, for example, in DendroTweaks, is basically one file that defines your morphology, one SWC file, and you will need some mod files which define your ion channels, and you can find some of these files online. There is a database which is called ModelDB, where you can find various models of ion channels. Okay, so now we will create, let's say from scratch, a model following this structure. So first we define a path to our model, and the first object that we should create is the model object. Basically this is the main, let's say the main interface. We work most of the time with this model object to modify our model, and to create an instance of this model class we need to provide the path to a folder. In our case it will be a path to this Park 2019 folder. And then basically from this moment you don't have to manage the files. DendroTweaks will do it automatically for you. So there are some methods to access the files in the folder. For example, you can see what SWC files you have available, and we will use one of these files here. So basically there is a method to load morphology from an SWC file. We just provide the name of this file. We don't have to specify, let's say, the path, the format. We just need to provide the name of this file here. And what DendroTweaks creates for us using this load morphology method is this representation with 3 network graphs, which represent the point tree, the section tree, and the segment tree. So now we have them available inside our model. We can access them like this, for example, model.pointTree. And we can visualize our cell using the plot methods which are available, for example, for the section tree. Here we see the neuronal morphology. We can select whether we want to annotate, for example, the sections with their IDs. And we can provide different arguments to this function to change the way how we visualize our model. Okay, so this, let's just run it. It is not very important for now, we can just skip it. Yes, also what DendroTweaks allows you to do is to analyze not only the activity but also the morphology of the neuron. You can do basic measurements for your cell. So for example, you can calculate the number of sections in your model, the number of bifurcations, number of endpoints, and so on. Okay, so next we want to update the segments that we have in our tree, and this segmentation is defined using a parameter which is called d lambda, and also the segmentation the optimal segmentation will depend on the biophysical properties of the cell. So we need to specify them first. We specify first the membrane capacitance and the axial resistance in our model, the passive properties of our model. And then we run this set segmentation method. So we can see that initially the model was created with 124 segments using some default parameters, but when we updated the values with something that we want, something maybe more biologically plausible, now we need a higher discretization to accurately capture the behavior of the cell, the activity of the cell. And then we can visualize specific sections. So before we had the entire tree. Now let's say we want to visualize one of the branches. So we can select a section first of all, using the section tree. We just access the section using its index. Let me plot it once again with the indexes. So now we want to visualize specifically this branch with index 22. So we go there and specify section 3. Basically it's a list of sections, and we can just use Python syntax to access the specific section. And now this section also has a plot method to visualize it. So we see the section and its parent section in 3 different projections, and we can also see the segmentation. So now this section is breaking into 7 segments. So these bars here, they represent the segments. All of them have equal length. And for example, if we modify this d lambda parameter, if we increase or decrease it, this will affect again the number of segments that we have in our model. So the spatial discretization with which we want to work. But usually the default value is 0.1, which gives a pretty accurate spatial resolution for most of the cases. Okay, so now we have our morphology. We have segmented it into segments. Now we want to add the mechanisms. And first of all, we should add some default mechanisms using this method. So basically what this recompile equals true means, as I said before, we use the mod files. We use the mod files to model the ion channels, and usually when you use, for example, Neuron, without DendroTweaks, you have to manually compile these channels to yes, you have to manually compile the code for these channels, but in DendroTweaks you just specify whether you want to or not do it, and it does it for you automatically. Okay, and now we also want to add other channels, other mechanisms that we have placed in the mod folder here. So all of them are now available for our model. We want to add them. Okay, so now you can see that for each channel we created a specific folder, and inside it has a copy of the original file, and also it has this folder with some C++ code that Neuron simulator will use to run the actual simulations. So you don't have to do it manually when you use DendroTweaks. You can just use this add mechanism method. So now we can see all the mechanisms that are present in our model, and we can see the parameters, the default parameters, which are specified in the mod file. So for example, if we open this kv mod file, okay, we cannot open it here in Colab, but basically these parameters, they come from the mod files. Yes, but so far, if we access again, if we access model.params, which is the place where all the parameters of our model are stored, we still see only the membrane capacitance and the axial resistance. We don't see any of these channels yet, because first of all we need to insert the channels to specific domains of our cell. So now by default DendroTweaks use the information from the SWC file to define the domains. Now we have the axon, we have the soma, and we have two types of dendrites. We have the basal dendrite, which is called just den. This is the default name which is used in Neuron. If you want, you can rename it using DendroTweaks. Also, we have this apic domain, which is the apical dendrite. Yes, and these domains, they correspond to the colors that we saw earlier when we visualized our morphology. So the orange here is the soma, and this is the axon, the yellow branch, and here we see the basal dendrites in green and the apical dendrite in blue. So we have these domains. We want to now insert the ion channels there. So we use this model insert mechanism method. to do it. And basically, in this case, for this specific model, we insert the same channels everywhere, but then we will change the distributions of these channels. Okay, this is just a way to see what parameters correspond to specific channels. So we, also one thing to mention here is that we have this pseudo mechanism which is called independent, and it collects some independent parameters which do not belong to any ion channel or some other mechanism. So here we have our capacitance, axial resistance, and we have also the equilibrium potentials for the ion channels, which are not specific, which are not bounded to a specific channel, but shared. For example, all potassium channels will depend on the equilibrium potential of potassium ions. But also we have properties, for example, that are specific for a given channel. For example, here for sodium channel, we have the maximal conductance of this channel and some other parameters which define its kinetics, the activation and inactivation curves that we saw earlier. Okay, and this is just another way to look at the parameters as a table. We see here again the mechanism, the parameter, and here we also see the groups and the distribution functions. And now we'll see how to modify these distribution functions, because now most of these values that we have here are just some default values collected from the mod files, but we want to actually create the distributions that are derived from some experimental data. So we want to specify where and how we want to distribute our channels. So for this we need to create the segment groups, and by default DendroTweaks creates for you the groups that align with the domains. So we have one group for the apical domain, one group, for example, for somatic domain, and we have a group which is called all, and it combines all segments together. So if you specify some parameters for this group, you will modify all the segments that you have in your model. So we have these default groups, but we also want to specify more specific collections of segments, more specific groups. For example, only for thin dendrites. And we can add a custom group to our model using this add group method. Here we specify the name of our group, we specify the domains in which we will be looking for the segments, and we have different options to select domains, to select segments from these domains. So in this case we will use the diameter, because we want to select only thin, only segments from the thin dendrites. So we specify section diameter here and the maximum value 0.8 micrometers. So every segment that has diameter larger than this value will not be included into this group. And we do something similar for thick dendrites. In this case, we specify the minimal value of the diameter. And another way to select segments could be, for example, the distance from the soma. So we can create a group which includes only the proximal part of the apical dendrite in this case. So every segment which is at a larger distance than 260 micrometers from the soma will not be included in this group. Okay. Okay, I should run this. So now we created these groups, these custom groups, and now we actually can modify the parameters of our model. So we use this setParam method here. We specify the name of the parameter and the value. This is the simplest case. If we want by default to modify a specific parameter everywhere in every segment of our model, we can just use this simple syntax here, just the name and the value. If we want to specify a group, we can provide it using the group name argument. And here we can use these default groups, or we can use these custom groups that we created earlier. In these groups, we just use a constant value. So for every segment in the dendritic group, for example, this parameter, maximum conductance for the KM channel, will get this value. So the distribution is kind of constant in this case. But we can also provide more parameters to this function, so we can specify the distribution type. So for example, for this, for one of the calcium channels, we want to use a linear distribution. So we specify it as the distribution type, linear, and we specify some parameters. So we specify the slope of this linear function and the intercept. So yes, so this is the general idea, to create groups of segments and to specify the distribution functions. Okay, here we just specify some extra parameters, not very important for now. And now we can visualize various parameters. So here we have the path distance from the soma. The colors indicate different domains. So, and here we have various sections. So for example, for this for the conductance of the leak channel, we have a constant distribution which is the same everywhere, because we just specified it using the name and just the value that we want. So here we see this value. When we created a more complex distribution of channels for the calcium channel, we now see this plot that shows us that the distribution is not uniform. So it is defined by, for example, for the basal dendrites, these green dots here, we see that it follows a linear function, which is increasing with the distance from the soma. And in this way we can replicate distributions of channels from the experimental data, for example. Okay. We also need to set some parameters for the equilibrium potentials for different ions that we have in our model, and we will specify the temperature and the initial voltage. So these are just some parameters that are used for the simulation. They are not related to any specific channel. And now if we again open our table with parameters, we can see that now these values are updated. Now for some of them we have for example linear distributions, or we have some sinusoidal distributions. So now these are not default values, these are the values that we specified in the previous step. Okay, and now to represent our biophysical parameters, we can generate the configuration file, the JSON file that I mentioned earlier. So we can find it here in our folder. Let me see if I can open it. Yes. And inside we see basically a description, a more compact description of what we have done so far. So we have here the mapping from the domains to channels. So we see what channels are inserted in each of the domains. We see the groups that we created before. And we see all the parameters that we specified. So for each parameter in the model, we have all the groups that we use to distribute this parameter, and we have inside these groups we have the functions that define the distribution. So this is a compact way, let's say, to store your data once you created your model, and basically then you can use you can share these files, and someone else can reproduce easily your model using this configuration file. And you can also reuse it later. So one straightforward application could be, for example, that you create your model using the code, and then you can use this file to open it with the graphical interface of DendroTweaks. Okay, so we have specified the morphological properties, the biophysical properties. Now the third component that we need is the stimuli, and we need also to specify some parameters of our simulator. So in this case we are using Neuron, so we delegate, let's say, the simulation to Neuron. And how it works, basically we specify, let's say, all the parameters, all the morphological data using our model object here in Python, and then when we want to run a simulation, DendroTweaks automatically creates a model in Neuron and runs a simulation for us, and we can record, for example, different variables from this simulation. So let's add one recording to the soma. So first of all, we will create this variable for the soma. Basically, it's the section in our section 3 with index 0. So it's the very first section in the list. And we want to add one recording for membrane potential at the soma. So we specify here the section that we want to use. We specify the normalized location along this section. So let's say it goes from 0 to 1, and 0.5 corresponds to the middle of the soma. And also we can record different variables. So by default we are recording voltage, so we can specify it, but it's also the default value, so we don't, if we want to record voltage, we don't have to specify it explicitly. So we created a recording, and now we want to also add current clamp. We want to inject a step current at the soma. So again, we specify the section, the location, and now we also pass some parameters of the stimulus that we want to use here. And now we are ready to run our first simulation. So we specify here time in milliseconds, and we run our model. Now it will take some time. Okay, we're ready. So now what we have done so far is that we created a model and we ran a simulation. We recorded activity from the soma. Now also DendroTweaks provides you with some functions to analyze the activity of your neuron. So first of all, you can see all recordings using model recordings. Yes, so we have, so basically it's a dictionary which contains recordings as vectors of values. So these are the values of the membrane potential. So if you need a specific recording, you can access it, for example, like this. Actually, you need to specify here the segment. Yes, this is the segment from which you record the activity. So it could be soma 0.5. Yes, and now we have this specific recording, but also you don't have to do it manually. You can just use some of the default functions that allow you, for example, to analyze the somatic spikes. So we import these two functions here, and we plot activity at the soma, and we can measure the number of spikes, the width of each spike on average, the amplitude, we can calculate the frequency, and so on. And now again, so we created, let's say, this virtual experiment. If we want to reproduce it later, we can store it in a file. So we click here, we call this export stimuli method, and we provide the name for this stimulation protocol. And if we go to the stimuli folder of our model, we can see these subfolders. So we have some of them that I have created earlier, but this is the one this is the experiment that we created now in this file, and it's inside it will have several files. So first of all, we have again a configuration file in JSON, but it is different. In this case, it just has some metadata and some simulation parameters. For example, the temperature, the initial voltage, the time step, and the duration of our simulation. So basically, everything that you need to recreate again the same experiment. And we have also these CSV files. So basically, these are just tables like that specify where we want to place our recordings. So they follow exactly the same logic as we used to add recordings. So we have, this is our soma, this is the index of the soma, which is 0. This is the location that we specify, and this is the variable that we recorded. In this case, it was voltage. So now if we want to reproduce again this experiment, DendroTweaks will just take the data from these tables and do it for us, so that we don't have to write the code again to reproduce our model. And we have the same for the current clamp, but here we have also the amplitude, delay, and so on. Okay, so this is the general idea. Yes, and now I will also show you how to add synapses. So first of all, to create, let's say, a new experiment, we want to remove all the previous stimuli and remove all the recordings that we have so far. In this case, we will add again the same recording as we did before, so we don't have to we didn't have to remove it actually, but it's better to clean everything before you start a new experiment. Yes, and now we want to add a population of virtual neurons. If you remember the slide from the presentation, for the presynaptic cells, we don't model them explicitly, but we will just specify some of the properties of this presynaptic cell. So we add a population which we'll call excitatory neurons, whatever, and then we want to specify the segments in which we want to place our synapses. So in this case, earlier I created this segments object using the group of apical segments. We specify also the number of synapses, and we specify the type, and we can modify different parameters. So for example, we can modify the activation of the synapses. So let's say they will be activated at 30 Hertz rate. And also we can modify the kinetic parameters. So if you remember before, we saw this curve, let's say, of the postsynaptic potential. We can modify in the rising phase, the falling phase of these postsynaptic potentials. Now again, we run our model with just some random synapses, random synaptic inputs. We can visualize the times at which the virtual presynaptic neurons were activated. Yes, and again we can export this protocol. So in this case we'll have also the file with all the synapses that we have in our model. So we'll have again the section and the location along that section where we want to place the synapse, and we will have also the population index. And if you see here at, in the JSON, we have also the populations. So this is our excitatory population that we created. It has index 0. So all synapses here, they belong to this population. And all of these synapses will take their properties from the values that are specified in this population. So if we want, we can easily reproduce this experiment. Okay. And so basically what we see here is another experiment where we measure the voltage attenuation in dendrites. And also we have an experiment to measure dendritic nonlinearity. So for now, I think we are done with this tutorial. I just want to briefly mention what else you can do with DendroTweaks before we go to the Q&A session. Okay. So yeah, so we have covered so far the basics of how to create, let's say, your first model, but also you can find many other tutorials in the documentation. So you can find more information about the SWC file, what exactly is the format. Again, there is a tutorial about distributing channels. You can find all possible distribution functions and so on. Also, you can find all the experimental protocols that you can use to validate your models. So we have covered some of them, but not all of them. And also there are some more advanced tutorials. Again, so one thing that you can do in DendroTweaks is you can modify the morphology. In our tutorial, we just used the morphology as it is, but if you want, you can remove some branches or modify some branches, for example. There is also a nice way to calculate the distances between different parts of the dendritic tree. For example, this can be used to analyze synaptic inputs, the distributions of synaptic inputs, and so on. Also, one thing that we haven't covered is morphology reduction techniques. So this is also part of DendroTweaks. You can simplify your model. So for example, you start with a detailed model, and then you can end up with a more simplified morphology, so you can merge some branches into a single section. And these models, they will be more efficient, but of course you will lose some details. But as you can see here, for example, the somatic activity in these models is approximately the same, so it depends on your research question, if you want to go to a more detailed level or a more simplified one. And also, one thing I want to mention is that there is a way to visualize channel kinetics and to work with these mod files that we discussed earlier. So we can visualize channel kinetics for every mod file that we have. You can check how to do it in this tutorial. And another thing that you can do is standardization. So usually these mod files, they come in all different flavors. The equations are not standardized, but using DendroTweaks you can make your life easier and convert everything to the same format. But all these techniques are more advanced. Unfortunately, we don't have time today to cover all of them in detail. So today my goal was to give you the basic idea of what you can do. And also, before we switch to the Q&A section, I will remind you that you can always use the graphical interface. So now that you have the understanding of how, let's say, you build your model, it should be easy to navigate the graphical interface because we follow the same logic here again. So for example, we select a model as we did using the code, and again we need the three components, the morphology. So we select one of the SWC files here, which appears in the graphical interface, and we can analyze different parts of the dendritic tree, so we can visualize these sections, the segmentation. This is, by the way, is the segmentation graph. So these two views, they are linked together. If you select something in the segmentation graph, you select the corresponding section, and vice versa. If you select a section, you see all the segments that belong to this section. So this is the morphology. Now we can load the biophysical configuration. By the way, this biophysical configuration is the JSON file that we created earlier that represents all parameters in our model. Okay, and let's also load one of the stimulation protocols that we created. So we use this one, current soma, the polarizing current at the soma, and we can see here the activity of our neuron. And one nice thing that the interface allows you to do is that you can work with your model in a more interactive regime. So for example, you can modify the values. If I decrease, for example, current here, you see that the cell is no longer active. If I increase it, so I modify the value here and I press enter. So I will increase it even more, and I hit enter again. Now we should see more spikes because we inject more currents. And in this way you can modify many other parameters in your model. So you can go to this tab with biophysical parameters, and here you can see all the channels inserted in the cell, and you can select specific channel, for example sodium. You can visualize the kinetics of this channel using the interface, and also here you see the distribution. So for this one we have higher value at the soma and lower value in the dendrites. And if you remember before, we had this for example Ka channel. So remember we created two groups of thick and thin dendrites, and basically this is what we see here. So the blue segments that you see here are the segments from the thick branches, and the red ones are from the thin dendrites. And we specified different values for these groups of segments. So one is higher, the red one is higher, the blue one is lower. So the idea here is that the interface allows you to do basically everything that we have done so far using the code. I just decided to follow the tutorial with the code because it gives you better understanding of how of the principles, let's say, of biophysical modeling. And I think now you can choose whether you like to use the interface or the code. I think now it's time for the Q&A section. You have a link. Probably we can duplicate it also in the chat. Can we send a link to the chat? No? Okay. Yes. Okay. So.
A great one on the go.
No, it's fine.
Have a dedicated link.
Yes, we do. Here's the link. We'll now also join. Can you try to join using the link? Stop the stream. Okay. Okay, so see you there. Now we will stop the live stream. I want to remind you also about our schedule for the next part. So now we'll have the Q&A session. Then we'll have a lunch break, and then we are looking to see you for the part 2 of our tutorial, where Mihalis will be presenting Dendrify. But for now, see you for the Q&A session in Google Meet.
Perhaps let the link on, the stream on, so they can
I can't keep it on.